In January 2026, the FDA approved the first human clinical trial designed to reverse aging in human cells. Not slow aging, not treat age-related disease. Reverse it. The scientist behind it is Harvard geneticist David Sinclair. The backers were Jeff Bezos, Sam Altman, and Brian Armstrong. Billions were poured into a single bet. Results are expected by early 2027, and what they reveal could change medicine forever, or prove we have been chasing a mirage. To understand how we might reverse aging, we have to change how we define it. For decades, science treated aging as an inevitable buildup of physical wear and tear, like a machine slowly grinding itself to dust. But a growing field of genetics suggests a different culprit: the progressive loss of cellular information. Inside every human cell, the DNA sequence acts as a permanent blueprint. But which parts of that blueprint are actually read is controlled by the epigenome, a system of chemical tags that turn specific genes on or off. As we age, these tags shift. The DNA code remains perfectly intact, but the cell loses its ability to read it correctly, forgetting its original identity and function. Harvard geneticist David Sinclair frames this as the information theory of aging. He compares DNA to a compact disc. The genetic code is the digital music encoded on the surface, while the epigenome is the optical reader. Over time, physical scratches accumulate on the disc, distorting the playback. But the underlying music remains completely intact. If this model holds, the instructions for youth are never truly lost. They are simply obscured. And if we can find a way to polish away the scratches, the cell might remember how to play the music perfectly once again. Translating this theory into actual biology requires a molecular toolset discovered in 2006 by Shinya Yamanaka. He identified 4 specific proteins, now known as Yamanaka factors, that can revert any adult cell back into an embryonic stem cell. But doing this fully is highly dangerous. If you completely reprogram a cell, it forgets its identity. A heart cell or a brain cell loses its specialized function, turns into a blank slate, and can rapidly form chaotic tumors called teratomas. To bypass this risk, researchers developed partial reprogramming. By using only 3 of the factors, OCT4, SOX2, and KLF4, collectively called OSK, and expressing them for a short, controlled window, cells undergo a rejuvenation process without losing their cellular identity. This approach yielded a major result on December 3rd, 2020. The cover of the journal Nature announced the milestone with the headline, "Turning Back Time." Inside, David Sinclair's lab reported that they had packaged these 3 OSK factors into a modified virus and injected them directly into the eyes of blind mice. The targets were retinal ganglion cells, the crucial neurons connecting the eye to the brain. Under the influence of OSK, the old damaged nerve cells did not turn into tumors or lose their identity. Instead, their epigenetic state returned to a youthful pattern. More importantly, the change was functional. The blind mice regenerated damaged optic nerves and actually regained their sight. This was not a minor laboratory signature, it was the physical restoration of lost vision. Measuring whether this biological clock actually rewinds in humans requires a molecular timestamp. Epigenetic clocks track biological age by analyzing DNA methylation, specific chemical patterns on the genome that shift predictably over time. The most famous, Horvath's clock, maps these changes across 353 genomic sites. But a shift in these molecular markers only matters if it connects to real, physical improvement. In January 2026, the FDA cleared an investigational new drug application for ER100, a gene therapy designed to test this concept in human patients. Developed by Life Biosciences, the treatment targets 2 devastating age-linked optic nerve conditions: open-angle glaucoma and non-arteritic anterior ischemic optic neuropathy, known as NAION. This is a localized trial in the eye, not a systemic whole-body rejuvenation therapy. Under the protocol registered as NCT 07290244, patients receive a single injection of an adeno-associated virus carrying the OSK reprogramming factors directly into the eye. Once inside the retinal ganglion cells, the treatment is tightly controlled. Patients take oral doxycycline for a 56-day window to activate the reprogramming genes. Once the doxycycline is stopped, the genes switch off, preventing the cells from losing their identity or forming tumors. Led by principal investigator Dr. Weiss, this Phase 1 trial is primarily designed to evaluate safety and tolerability. While researchers will monitor visual acuity and blood-based epigenetic biomarkers, the core objective is ensuring the therapy does no harm, and establishing the baseline before epigenetic restoration can ever scale to the rest of the body. Life Biosciences is translating partial reprogramming from laboratory theory into clinical trials. Following the January 2026 FDA clearance, the company moved rapidly. On June 9, 2026, doctors administered the first dose of ER100 to a patient, officially starting the first human test of cellular rejuvenation. Initial safety and exploratory data are expected by late 2026 or early 2027. This milestone energized a growing, high-stakes industry funded by some of the most prominent figures in technology. David Sinclair, a founder of Life Biosciences, captured the weight of this moment during his address at the World Government Summit in Dubai on February 5, 2026, stating, "We are about to test for the first time in history whether we can reverse aging and cure diseases. The pursuit has attracted billions of dollars to rival ventures. Altos Labs, backed by Jeff Bezos, launched with approximately $3 billion to focus on systemic cellular rejuvenation, bringing in pioneers like Shinya Yamanaka and Steve Horvath as advisors. Sam Altman committed $180 million to Retro Biosciences, which explores cellular reprogramming alongside other age-related therapies. Meanwhile, New Limit, backed by Coinbase founder Brian Armstrong, secured a $130 million Series B round to target epigenetic reprogramming. This massive influx of private capital has dramatically accelerated research timelines, yet industry analysts point out that financial backing is not scientific validation. While billions of dollars can build state-of-the-art laboratories and hire expert researchers, they cannot alter the rigorous biological outcomes of human clinical trials. This early trial represents a highly localized intervention, not a proven blueprint for resetting the entire human body. Many geroscientists, including João Pedro de Magalhães, point out that correcting a specific optic nerve pathology looks more like targeted tissue repair than systemic age reversal. Organism-wide aging involves complex metabolic and inflammatory pathways that a localized ocular injection simply cannot address. The clinical pathway from animal models to humans is notoriously steep. Therapies that succeed in mice or primates routinely fail in human trials. Because the current study is designed strictly to evaluate safety and tolerability rather than efficacy, any claims of imminent whole-body rejuvenation remain speculative. Restoring function to a single eye tissue is a significant medical step, but it is still miles away from reversing biological sickle age across the entire human system. Subscribe to follow this scientific development. If ER100 proves safe, the development path will progress tissue by tissue, rather than through immediate, whole-body rejuvenation. Kidneys, the liver, and the brain represent the next target classes for epigenetic restoration. Success would shift the focus of medicine toward treating aging itself as a clinical condition, rather than managing separate late-stage diseases. Meaningfully extending healthy human lifespan carries immense social and economic implications for global workforce productivity. This future sits at the center of David Sinclair's prediction that modern healthcare could look entirely obsolete within 10 to 20 years. Yet this timeline remains a long-term forecast rather than a guaranteed clinical outcome. We stand at the absolute frontier of medicine, where aging is treated not as fate, but as a biological glitch. If science finally unlocks the code to turn back your biological clock, would you choose to reset it? Subscribe for more deep dives into the future of science.